Chimeras. Right now, in laboratories across the world, scientists are growing human organs inside pigs, not transplanting them, growing them from scratch. These aren't science fiction experiments, they're real, they're happening, and they're called chimeras. The term comes from Greek mythology, that fire-breathing creature with a lion's head, goat's body, and serpent's tail. Here's how it works. Scientists inject human stem cells into pig or sheep embryos just days after fertilization. These human cells integrate into the developing animal, growing alongside pig or sheep cells. The goal is to create animals with human organs that can be harvested for transplantation. We're talking about pigs with human pancreases, sheep with human livers, animals that are maybe 1% human by cell count, but potentially carrying fully functional human organs inside them. This technology exists because we have a massive organ shortage problem. Over 100,000 Americans are waiting for organ transplants right now, and about 17 people die every day waiting for organs that never come. If scientists can grow human hearts or kidneys inside pigs, it could save countless lives. But here's where it gets complicated. What happens if those human cells don't just form organs? What if they migrate to the animal's brain? The ethical questions are massive. At what point does adding human cells to an animal create something that deserves different moral consideration? Most countries have strict regulations. Research is limited to early-stage embryos, and these chimeric animals are usually destroyed before birth. Japan recently lifted its ban on growing these chimeras to full term, which means we might soon see pigs with human organs walking around research facilities. Scientists in China claim they created monkey embryos containing human cells that survived for nearly 3 weeks. The closer we get to primates, the more uncomfortable this becomes. These aren't hypothetical what-ifs anymore. Cybrids. If chimeras weren't weird enough, let me introduce you to cybrids. The name stands for cytoplasmic hybrids, and they're arguably even stranger than chimeras. Imagine taking a cow's egg, removing all the cow DNA from it, and then injecting human DNA inside. What you get is 99.9% human genetically, but it's growing inside what used to be a cow egg. That's a cybrid. British scientists created the first human-cow cybrids back in 2008, and they weren't trying to make headlines. They were trying to solve a problem. Human eggs are incredibly difficult to obtain for research. They're scarce, the donation process is invasive, and there are huge ethical concerns about using human embryos for experimentation. But cow eggs? Those are abundant and cheap. The process is weirdly simple on paper. You take an unfertilized cow or rabbit egg and remove its nucleus, which contains all the animal's DNA. Then you inject a human cell nucleus into that empty egg. The egg provides the machinery to start cell division, but the DNA is human. These cybrids develop for a few days, just long enough to produce human stem cells for research, and then they're destroyed. By law, they can't be allowed to develop beyond 14 days. Here's what's wild about this. These embryos are human where it matters for research, the DNA, but they're housed in animal cellular machinery. Some people argue this is less ethically problematic than using human eggs because there's no human egg involved. Scientists in the UK have created thousands of these things, using them to study conditions like Parkinson's disease and motor neuron disease. The technique works with multiple species too. Human rabbit cybrids have been created, and theoretically, you could do this with almost any mammal egg. And while these entities are destroyed before they develop into anything recognizable, the fact that we can create them at all shows just how blurry the line between species has become in modern biology. Humanzee. In 1926, a Soviet scientist named Ilya Ivanov tried to create a human-chimpanzee hybrid. Not as a thought experiment, he actually tried to do it. And the truly disturbing part is we're still not entirely sure what happened. This is one of those historical experiments that sounds too insane to be real, but the documentation exists, and it's nightmare fuel. Ivanov was a world expert in artificial insemination. He'd successfully created hybrid animals before, zebra horse hybrids, and bison cattle hybrids. And he thought the same techniques could work with primates. He pitched the idea to the Soviet Academy of Sciences, and Stalin's government actually funded it. The goal was partly scientific curiosity and partly ideological. They wanted to prove that humans were just animals, and that there was no divine spark separating us from other species. So Ivanov traveled to French Guinea in Africa with his son and set up operations at a primate research station. The plan was to artificially inseminate female chimpanzees with human sperm. He attempted this at least 3 times with chimps at the station. None of the attempts resulted in pregnancy, or at least none that we know of. When that failed, Ivanov proposed the reverse experiment, inseminating human women with chimp sperm. This is where things get murky. There are reports that he found volunteers, women who agreed to the experiment, but before anything could happen, Ivanov was arrested during Stalin's purges in 1930. He was exiled to Kazakhstan, where he died 2 years later. The research was abandoned, and most of the records were destroyed or classified. From a genetic standpoint, it theoretically could have worked back then. Humans and chimpanzees share about 99% of our DNA, and we diverged from a common ancestor only about 6 million years ago. Some scientists think a hybrid might be biologically possible, but it would face massive developmental problems. But the chromosomes wouldn't match up properly. Humans have 23 pairs, chimps have 24, and even if fertilization occurred, the embryo would likely miscarry. The ethics of even attempting this are so far beyond acceptable that no legitimate scientist would touch it today. But Ivanov tried it anyway, and somewhere in Soviet archives, there might be records of what actually happened. Humanized pigs. Pharmaceutical companies are raising entire herds of pigs that produce human proteins in their blood. These aren't just carriers of human organs, these are pigs that have been genetically engineered with human genes to manufacture human medicines inside their bodies. They're living pharmaceutical factories, and they're producing some of the most expensive drugs on the market. The process starts with genetic engineering at the embryo stage. Scientists insert human genes into pig embryos, specifically genes that code for therapeutic proteins humans need. When these pigs mature, their cells read those human genes and produce human proteins. The proteins circulate in the pig's blood, or get secreted in their milk, and then pharmaceutical companies harvest them, purify them, and turn them into drugs. One of the most successful examples is human antithrombin, a blood-clotting protein. People with antithrombin deficiency face life-threatening complications during surgery. A company called GTC Biotherapeutics created goats with human antithrombin genes, and those goats produce the human protein in their milk. The same technology has been applied to pigs for different proteins. What makes pigs particularly useful is their size and reproductive capacity. A pig can produce much more blood or milk than a mouse, and pigs reach maturity faster than cattle. You can establish a herd of these humanized pigs relatively quickly and get industrial-scale production of whatever human protein you've engineered them to make. It's more efficient than growing the proteins in cell cultures, and for some complex proteins, it's the only way to produce them correctly. By the way, if you're finding these experiments as disturbing and fascinating as I am, I post content like this regularly. I'd appreciate if you subscribed. Thanks. Xenotransplant baboons. In the 1980s and 90s, surgeons attempted something that shocked the medical world. They transplanted baboon organs into human patients, not mechanical devices, actual baboon hearts and livers, beating and functioning inside the human bodies. Most attempts failed within hours or days, but one patient, a baby known as Baby Faye, lived for 21 days with a baboon heart pumping her blood. Baby Faye was born in 1984 with a fatal heart defect. No human donor was available, and her parents agreed to an experimental xenotransplant. Surgeon Leonard Bailey transplanted a baboon heart into the infant, making her the first human to receive a primate organ transplant. For 3 weeks, that baboon heart kept her alive. It beat, it circulated blood, it functioned as a heart should. But ultimately, her body rejected it, and she died. The procedure was controversial even then. Critics argued that baboons were too evolutionarily distant from humans for the transplant to ever succeed long-term. The immune rejection would be too severe. Bailey believed that infant immune systems might be more tolerant of foreign tissue. Other attempts followed. In 1992, a man received a baboon liver transplant at the University of Pittsburgh. He survived 70 days before dying of infection. These weren't technically creating hybrid organisms because the organs remained baboon organs, but they created hybrid physiologies. Human blood flowing through baboon organs, human immune systems trying to tolerate primate tissue, human bodies dependent on animal biology to survive. The experiments eventually stopped because the rejection problems proved insurmountable with available technology, but they paved the way for modern xenotransplant research. Today, scientists are genetically engineering pigs with human genes to make their organs less recognizable to the human immune system. In 2022, surgeons successfully transplanted genetically modified pig hearts into brain-dead human patients, and the organs function for days. Humanized monkeys. Chinese scientists have created monkeys carrying human brain development genes, and those genes made the monkeys smarter. This isn't speculation. The researchers tested the animals and documented improved cognitive performance compared to normal monkeys, We're talking about primates that are already intelligent becoming more intelligent because of human DNA. The experiment involved inserting human copies of the MCPH1 gene into rhesus macaques. This gene plays a crucial role in human brain development and is one of the genes that differs most between humans and other primates. It affects how brain cells divide and organize during fetal development. The transgenic monkeys were born, raised, and tested on various cognitive tasks. The results were unsettling. The humanized monkeys showed better short-term memory than control monkeys. They performed better on tests requiring them to remember colors and shapes. Their brains also developed differently, taking longer to mature, which is actually a human trait. Human brains develop more slowly than other primate brains, and that extended development period is thought to contribute to our enhanced cognitive abilities. The research sparked massive ethical outcry, even within the scientific community. Creating animals with enhanced intelligence, especially primates that are already cognitively complex, crosses a line many people find unacceptable. If human genes can make monkeys smarter, where does that stop? What if you added more human brain genes? What if you did this to apes, which are even closer to humans cognitively? China has continued this line of research despite international criticism. They've created multiple generations of these transgenic monkeys and expanded the work to include other human brain-related genes. The stated purpose is to create better models for studying human brain disorders like autism and schizophrenia. We've demonstrated that we can alter the cognitive abilities of our closest relatives by inserting human DNA. Human blood sheep. Scientists in Nevada created sheep with human blood. Not blood cells transfused from humans, but blood that the sheep's own body produced using human genes. These sheep had up to 15% human cells in their circulatory systems, and those human cells were making human hemoglobin and human blood factors. The research was conducted at the University of Nevada using fetal sheep as hosts for human stem cells. Researchers injected human hematopoietic stem cells, the cells that produce blood, into sheep fetuses while they were still in the womb. Because fetal immune systems aren't fully developed, the sheep didn't reject the human cells. As the sheep matured, those human stem cells took up residence in their bone marrow and started producing human blood cells alongside the sheep's natural blood cells. The result was sheep with chimeric blood, a mixture of sheep and human red blood cells, white blood cells, and platelets all circulating in the same cardiovascular system. The human cells function normally, carrying oxygen, fighting infections, and clotting wounds, just like they would in a human body. But they were doing it inside a sheep, supported by sheep organs and sheep physiology. This research was aimed at understanding how human blood diseases develop and testing potential treatments. By creating sheep with human blood systems, scientists could study human blood disorders in a living organism without using human subjects. They could infect the human blood cells with human-specific diseases, test drugs, and observe long-term effects in ways that would be impossible with human volunteers. What makes this particularly strange is that blood is so fundamental to identity. Your blood type, your immune markers, your blood chemistry, these are defining characteristics of who you are biologically. These sheep had human blood coursing through their veins, human immune cells patrolling their bodies, human biology operating inside animal hosts. Humanized rats. Researchers have created rats with human immune systems, and these rats can catch human diseases that normal rats can't. They get infected with HIV, develop human-like immune responses, and serve as living models for testing treatments. These humanized rats are genetically rats, but immunologically, they're responding to diseases like humans do. The technology uses immunodeficient rats that are bred to lack their own immune systems. Scientists then inject human immune cells or transplant human thymus tissue into these rats. The human immune cells colonize the rat's body, taking up residence in the spleen, lymph nodes, and bone marrow. They start producing human antibodies, human T cells, and human immune responses. The rat becomes a host for a functioning human immune system. This is medically crucial because many human diseases, particularly viral infections, don't affect normal rodents. HIV is a perfect example. The virus specifically targets human immune cells and can't infect rat cells. But a rat with human immune cells, that rat can be infected with HIV, and the virus will replicate, attack the human immune cells, and progress just like it does in human patients. Suddenly you have an animal model for a disease that previously couldn't be studied in rodents. The same technology works for hepatitis, Epstein-Barr virus, and other human-specific pathogens. Researchers can infect these humanized rats, test antiviral drugs, study how the immune system responds, and develop vaccines. The rats suffer through human diseases, experiencing immune suppression and infections that their rat biology never evolved to handle. But the medical knowledge gained has been substantial. What's particularly eerie about these rats is that they can develop human immune responses to things in their environment. If you expose a humanized rat to pollen or peanut protein, it might develop human-style allergies. Its body, run by human immune cells, reacts the way a human body would. These rats are experiencing aspects of human biology, human susceptibility to disease, human immune dysfunction, all while being rats in every other way. If this exploration of human-animal hybrids sparked your curiosity about the weird edges of science and biology, consider subscribing so you don't miss future content. Thanks for watching.